In this tutorial, we will discuss about phosphorescence and fluorescence. In nature, some creatures can experience what we call biofluorescence. It is when a living organism absorbs light, usually blue or UV light, and then re-emits it as a different color, such as this fluorescent moray eel at Sakatia in Madagascar. And this is what fluorescence and phosphorescence is about. We will explain the physical principle of these two concepts and the difference. So fluorescence and phosphorescence are two types of photoluminescence. When we shine light onto an object that has this property, its molecule absorbs light and gets to the excited state. In this diagram, this excited state is at a higher energy. So this is the excited state, where this is the ground state. But this excited state is not a stable one, so the molecules will go back to the ground state to reach stability by emitting light in forms of fluorescence or phosphorescence. The difference between the two is that fluorescence is immediate, while phosphorescence is delayed and can last a little longer. So what does this mean in reality? Example of application is glow in the dark product, such as for light festival or party. So this is an example of phosphorescence. That is like in the image on the image of a woman on the left, while fluorescence has extensive application in markers for bioimaging, where fluorescent dyes are used to paint or bend to pens or bound to some biological sample so that it can be imaged using fluorescence microscopy. So the idea in fluorescence microscopy is that different parts of a biological sample can be painted differently using different fluorescent labels so that we can then distinguish, for example, microtubules from nuclei, from cell membrane, and so on. And so this is an example of an image that was taken from a fluorescence microscope. And the fluorescent dye will require higher energy to absorb than what it can emit. So the excitation wavelength of light to excite a molecule is lower than the emission wavelength, and the difference between the peak of the excitation and the emission spectrum is called Stokes shift. And this Stokes shift is very important in fluorescence microscopy to distinguish the fluorescence signal from the sample. So here, for example, we have this is the peak of the excitation with spectrum, where this is the emission spectrum. So now, as a part of experiment, to experiment this phenomena, we use a board that have been painted with phosphorescent paint. The paint contains photoluminescent pigments. The first experiment that the student can then test is to use a green laser to draw on the board. So there's the board. They should be asked to observe what they see. Do the same experiment with a blue laser and let them observe the difference. They should see that they can actually write with the blue laser on the board and not with the green laser. There is an explanation to this. The pigments on the board has an emission wavelength in the green range. To be able to emit light in the green region, it needs to absorb higher energy than what it can emit, similar as what we discussed earlier. The blue laser has higher energy than the green laser, so the blue laser is expected to contain enough energy to excite the pigments and emit phosphorescent light afterwards. As this activity requires the use of a laser, it is mandatory to instruct students about laser safety. Once again, a laser is not a toy. Given that the laser light is directional, if it is concentrated, its energy can be very high. It can damage the skin and the eyes. So it is therefore important to remember to never direct a laser light to our own or someone else's eye. It also is important to be mindful of any potential reflections of surface. And that concludes this tutorial.